Hi, so today we're going to be talking about pharmaceutical drugs. We're going to talk about the history of them, we're going to talk about the thinking behind how they were developed, and we're going to talk about some modern examples as well. Okay, so let's start our conversation about drugs and drug molecules with the idea of natural products. These are molecules that are derived from natural sources, and especially plants. To give you a good example, so willow bark, you can take off parts of willow bark, and then you actually can extract, or you can remove a biologically active compound, salicylic acid, and this salicylic acid has been known for quite a long time to be a fever reducer. For the plant, the plant uses it as a plant hormone, but for us, it has this anti-inflammatory kind of action, and it's been listed by the World Health Organization as one of the essential medicines of a developed country. As I said, it's been used for quite a long time. So Native Americans, it's been shown in archaeological finds that people have been using it for hundreds, if not thousands, of years to help out with inflammation. One of the problems with salicylic acid was that if you just ate a whole bunch of salicylic acid, it was pretty acidic. And if we remember functional groups, we have both a carboxylic acid here, and we also have this alcoholic group, and they're both acidic. And it tended to cause these stomach ulcers and this intestinal bleeding if you ate a lot of it, and so people liked this compound because it reduced inflammation, but it also had these terrible side effects. And so Bayer at the time, a company in Germany, they really wanted to find a derivative that they could make that would not have these side effects, but still have the good effects, the intended effects. And Felix Hoffmann, a chemist in that company, really made his mark, and he was able to synthetically derivatize a salicylic acid to make acetylsalicylic acid, also known as aspirin. And this was one of the first great discoveries of developing a novel pharmaceutical drug, and this is based upon a natural product. And actually, developing pharmaceuticals off of natural products still happens pretty regularly today. Up to 70% of all of the pharmaceuticals on the market right now are in fact derived at some point or have some kind of derivation from a natural product, typically a plant or a microorganism. Not all drugs are originally derived from natural products, though, and some are entirely synthetic. So some are made entirely in a chemistry lab. And to understand this story of how chemists began to think about making purely synthetic drugs, we have to start out with Perkin's mauve. So Perkin, as a young person, you think he was in his 20s, was successful in developing a stable, really lustrous, really bright purple dye. He did this in 1856, and it was so popular that it actually it generated this massive chemical dye industry. So up to this point, there hadn't been anything that had been stable that people could dye their purple dresses with, except for a really rare seashell that it could be crushed up. So up to this point, people really couldn't use the color purple in clothing industry, and so this is actually an original dress from the 1860s that Perkin dyed using his method. And with the advent of all these new dyes that were being generated, some scientists had some thoughts. And one of them was Paul Ehrlich. So Paul Ehrlich was a biologist, and he ultimately founded a diverse number of fields, hematology, immunology, antimicrobial chemotherapy, but he did so because what he was thinking of was, well, okay, we have these dyes, these dye molecules, and these dye molecules have these vibrant colors, and they can dye clothing. Can they also dye parts of the cells? And indeed they could. So you can see here, these are stains that stain the nucleus of a cell specifically. And so Paul Ehrlich had an idea, and that was, well, you know what? If a dye molecule can specifically stain or specifically interact with one part of the cell and not the other, perhaps certain molecules might be able to kill certain cells and leave other ones the same, or healthy. And so this was his idea. It's called a magic bullet idea. And at the time, he wanted to treat a scourge of syphilis that was happening in Europe. And so the idea was he wanted to develop a molecule that could potentially kill the syphilis and not kill the people at the same time. And he came up with these interesting arsenic-containing molecules that did show that he could actually kill syphilis and not kill the patients. And for this work, this idea, he actually received the Nobel Prize in 1932. Another contemporary of Paul Ehrlich, Gerhard Domag, took this idea and took it even further. So Domag was a medical student in World War I, and he served in these hospitals in Russia, and so he got a real face-to-face kind of view of what disease can do to people. And so he kind of came back from the war, and he spent all of his time researching antibacterial drug development. And up until this time, there really was not anything that would be antibacterial. So if you got a bacterial infection, you just had to hope that you didn't die from it. And so what he did was he went and he found this large library of dye molecules, and he began testing as many dye molecules as he possibly could to see whether or not any of them would potentially kill bacteria, and he actually was successful. So Prontosil rubrum, which is this molecule right here, Prontosil rubrum is a red dye, and it apparently would kill streptococcal infections. In fact, we now later learned that it's not in fact the dye, but the dye actually gets broken down into a new molecule called sulfanilamide. Sulfanilamide is a class of the sulfa drugs, which are even still used today. And these are very effective drugs against strep infections. And for this, he was awarded the Nobel Prize in Medicine for developing the very first antibiotic in 1935. Okay, so the question might be, how do these drugs work? So how does a sulfa drug work? How does anything work in the body? And the answer is that for the most part, most drugs are targeting a protein or a protein complex. So if you remember, proteins are these windy, twisty polymer of amino acids, and they all have different jobs. So some are structural, some carry things around, some have to do with your immune system. A lot of them are enzymes, so a lot of them catalyze reactions. So they all do something. And so a lot of times disease states, whether it's a bacteria or it's something going on in your own body, it has you want to potentially target a protein to either turn a necessary protein off in a microbe, so maybe the microbe needs this protein to survive, or if it's some sort of abnormality in a person, you can affect maybe their health by targeting a protein in their body. So you have a drug molecule, and this drug molecule is a key, and the key can fit in maybe the lock space of a particular protein. And so drug chemists will design the shape of a molecule to fit nicely into that protein, and effectively, once the key is in there, it's stuck and it won't come back out again. And so this drug target complex, it kind of shuts the protein down. Its active site has been turned off, and so that's the idea. And of course, some drugs work more effectively than others, but chemists attempt to manipulate the efficacy of the drug simply by changing the shape, changing the functional groups of the molecule until you get the desired effect. Is it tough to make a new drug? The answer is extremely tough, actually. While I'm certainly would never say that the pharmaceutical company deserves our pity, because they're not always the most ethical of all the industries. They do have a really challenging job ahead of them to develop new drugs. So the drug discovery process typically takes between 10 to 12 years. So and they'll screen thousands of different molecules. So they look at thousands of different molecules. Ultimately, they center it onto some of the most efficacious molecules, so some of the best working molecules, as they go into the clinical trials, where they go through testing it in people ultimately. And after maybe 10 years, the FDA will approve one of their drugs. And currently, though there's estimates that are much higher than this, and there's some that are a little bit lower than this, most people agree that the average cost to develop one new drug sits at around $2.6 billion currently. And this comes down to that you have in drug development, you have a lot of failure. So most of the time, you're trying to develop compounds that simply do not work. That we simply do not understand enough of the biochemistry yet and the biology yet to get everything right, and a lot of times things will fail pretty spectacularly in the clinical trials. But when you finally get one approved, then typically pharmaceutical industries will charge quite a bit for it to recoup all of their losses. A great example of a modern drug might be one of the statins. This is atorvastatin, also called Lipitor. So Lipitor is really good at lowering cholesterol levels. All the statins are. This, believe it or not, actually was originated from a natural product, lovastatin. Lovastatin is isolated from a yeast on bean curd, and people found out that the lovastatin would reduce people's cholesterol levels, and so they chemically modified this particular molecule until they came up with a more effective Lipitor, the atorvastatin molecule. And what this effectively does is it binds to an important enzyme in your liver, which basically biosynthesizes cholesterol, and it shuts down the whole pathway, so your body can't really make very much cholesterol anymore. Lipitor used to be the top-selling drug in the world, global sales of about $9.6 billion per year. But as with all drugs, they only have about a 20-year composition of matter patent, which means that even though it takes them 12 years to make it, they only have about 8 years on the market before it goes off patent and generics can take on. So at 2011, this patent ran out, and then the generics kind of took over, and sales fell pretty precipitously in the next year to about 3.9 billion, which is nothing to sneeze at, but it's but it was quite a hit to Pfizer.